Let's approach protein structure, shall we? So it's going to be based around one of the things that I like, which is the structure of the SARS coronavirus 2 spike protein. This is the thing you've probably been vaccinated against. Call it the COVID vaccine. Yeah, that's fine. All right, so let's talk about this thing. And you can see we've got a nice little animated picture of a rotating protein molecule. Look at that. Isn't that beautiful? I think it is. Okay, so as you know, there are many different amino acids. Here they are once again. So, what's in a protein? Well, it can be a range of things, but when you get to a large protein, and the spike is large, 1,255 amino acids. Wow! Think how long that gene would have to be. 3 times as long as that. 37, 3,800, yeah, nucleotides. Long gene, very long. So here we go. We've got all these with the charged positive or negative side chains. Some of the special cases, so glycine and proline tend to be in loops. Cysteine tends to be one of the attacking amino acids. And then serine and threonine are also going to be quite attacking quite often. And you got your hydrophobic guys down here. You're going to find those, you'll tend to find those in the center, like the core of a protein, where they can interact with each other and they don't have to participate in hydrophilic interactions. Good for them. None of these things are absolute. You can find any of these amino acids anywhere a little bit. It's just that they have tendencies. These are the tendencies. So in the SARS spike, you actually find a little bit of all the amino acids, all 20 of the common ones, but in different proportions. Yeah. That's true of pretty much any protein. You'll have different proportions that do different things. But that's not what we're here to talk about. We're here to talk about, like, okay, what's a protein? How do I make a protein? How do I understand it? Well, protein's a chain of amino acids. So shown here are a bunch of amino acids. So Q is glutamine, the Q-tamine, yeah. A for alanine, P for proline, etc. And so if we were to lay these all out in a row, it would look like this. So this is an example of an amino acid sequence. Yeah, and you can tell it's amino acids and not nucleotides because there's no nucleotide K or L or E. Yeah. Okay. And so to turn these amino acids into a protein, which is something the ribosome is normally going to do, we're going to have an attack. We'll have this oxygen basically attacking that nitrogen, or I forget if it's the nitrogen that attacks. I think it's the oxygen, though. And so we will form a covalent bond between those, and that is the peptide bond between, this carbon and that nitrogen. This OH and one of these hydrogens is going to get kicked off. And they're going to form a water molecule. And so we call this a dehydration because we're going to squeeze out a water molecule's worth of atoms in forming this bond. Yep. And so we'll do that for every single one of these. So we're going to go that carbon over here, and this OH is going to be gone. The OH in this position is going to go away every single time. When they're all joined together, we can show them several different ways. So we can mark each amino acid in a different color. And so you can see the backbone and the side chains doing all their things here. Or we can just show the backbone. And so we'll ignore the side chains, but from this you can see that the backbone is making a twisty shape, which is an alpha helix. Yeah, you see that? Yeah, a little alpha helix. Normal, right-handed alpha helix. And so here we've labeled the different atoms in different colors. And these are the atoms of the backbone. So we have our nitrogens, carbons, and oxygens. All right. But there are different ways to visualize a protein. So what we've got going on up here and what we've got over here is called a ribbon diagram. And so a ribbon diagram is going to show us wherever there is a beta strand, and so look at all these beta strands up here. It's going to show us where there's an alpha helix, because it's going to be like this. The beta strand's usually going to have an arrowhead, and it's going to look like a ribbon. The arrowhead will show you which direction it's pointing. Like there you can see the red arrowhead going this way to the left. On a helix, they don't normally have the arrowhead. You can figure out the direction of the chain from the beta strands if there are any. So yeah, they're different models, but they're all showing the same molecule. And so here it is with its side chains on, and so we've marked the different amino acids. When there's two that are close together, I've put them together, but they're just attached by normal peptide bonds. Down here is a thing that we can call a space-filling model. So this is imagining a little sphere around every atom, and you can think of that as like the farthest extent that its electrons are allowed to go. It's not as precise as that, but that's reasonable. And so it's like this is the outer surface shape of the molecule. And so molecules, proteins, fit together, and substrates fit into proteins just like little puzzle pieces. And this is kind of the shape that the puzzle pieces are seeing as they're fitting in, or at least this is an easy way to visualize it that I think makes sense to people. Yeah. Because that's who we are, right? Yeah. Okay. And so, yeah, like we said, we're going to use these words. So what are we looking at over here? So primary, secondary sequence, primary, secondary structure. Here we got primary structure, which is our amino acids in the order in which they go. And here they're sort of twisted into a kind of a secondary structure. We're not showing the hydrogen bonds, but if it's in a ribbon diagram, if it's hydrogen bonded like an alpha helix, then it will show up as an alpha helix. Same thing with beta strands up here in the top. So secondary structure is going to be our alpha, beta, and everything else. Tertiary structure is going to be the overall fold. So I like the space-filling model for tertiary structures. I think that gives you kind of the shape. And what you can also do is put an electrostatic coloring scheme on that, which will show you where the positive and negatively charged parts, or partial positive and negative charges, are going to be located on the molecule. Because molecules are not just going to interact with shape matching shape, they're going to interact with plus and minus interacting, or with hydrophobic and hydrophobic interacting, drawn together by their mutual hatred of water. Yeah. Or I guess fear is the literal translation, hydrophobic. And then I think that space-filling models are also very nice for understanding quaternary structure, how multiple protein chains can come together to form a larger unit. Yeah. So for example, in the coronavirus spike, there are 3 chains that each have a primary structure that's a little less than 1,300 amino acids long. They fold up into a bunch of secondary structures. So the front of it is mostly beta strand, and the back half of it is mostly alpha helix. And then those secondary structures fold up into a shape, which is kind of like a lollipop, kind of that spike shape you've seen. But it takes 3 of those to make an actual spike molecule. So 3 of those chains. And so they make the actual, boom, quaternary structure that everybody's so familiar with now. There you go. And so this is our standard example NSP15, primary structure, secondary structure, the alpha and beta, tertiary structure, which can be divided into multiple protein domains. Here's one domain, second domain in the middle, and third domain at the end. And then the quaternary structure is how those domains, which are part of the same molecule, fold up. So if we're talking domain structure, this is like part of the tertiary structure. It's an element that makes up the tertiary structure. You can divide tertiary structure into multiple domains sometimes. And division into domains is kind of arbitrary because, as you remember, there are those different kinds of domains. Domains which, these are enough parts to do a function. Domains which we can kind of draw a circle around their structure and they seem to be internally hydrogen bonded. And domains that appear to be conserved, that appear to be in every instance of this protein that exists in nature, versus the area around them, which tends to be flexible. So there you go. All right, so that's another look at protein structure. And so just to review, so we're looking at primary structure here. And how can you tell? Because these are the amino acids. There's 20 different letters that can be amino acids, and well beyond A, C, G, and T, which would indicate a nucleotide sequence. How do we tell we're looking at secondary structure here? Well, it's got alphas, that's that curly little fish looking letter, and it's got betas, which is like a bee but with a long tail, just like that. There you go. Now you know Greek letters. You probably knew those already, though. This is a tertiary structure, yes, but what type of tertiary structure? What type of diagram would we call this, or representation? So let's look at it. Do you see any side chains? I do not. I only see backbone. I'm like, okay, so we've got backbone. Is the backbone showing where the alpha helices and beta strands are? And are they shown like flat ribbons? If they are, then that's what we call a ribbon visualization, a ribbon diagram of a protein. Yeah. And what's this? You can call this like an isosurface, or you can call it a space filling or three-dimensional, but this is basically the shape of the electron clouds around the edge of the protein. It's actually showing what its three-dimensional shape looks like. In this case, they've colored it so you can see the different parts which were important for the reasons covered in that paper. But there's different ways you can color this that are useful for other types of projects. So there you go. Protein structure and how we visualize it. I hope that makes sense. All right.